Ribosomes, given by George Palade in 1953. Ribosomes are ribonucleoproteins made up of RNA plus proteins. Ribosomes protein factory, synthesis of proteins, two subunits, small subunit, large subunit. Prokaryotes and eukaryotes both have ribosomes. Ribosomes is non-membrane bound organelle. Then in prokaryote it is 70 years, in eukaryotes it is 80 years, in mitochondria and chloroplast it is 70 years. 70 years subunits are 50 years and 30 years, and 80 years, that is in eukaryotes it is 60 years and 40 years. And S stands for sedimentation coefficient, indirectly a measure of density and size. In nucleus, right, the it has nucleolus that synthesize the rRNA, right, and there is movement in both directions through the nuclear pores, that is movement of proteins and rRNA. Okay, it's written in here like nuclear pores are the passage through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. That's it. The nuclear matrix is nucleoplasm, right? Telocentric chromosome has a terminal centromere. It means it has no, right? It has no chromatin on one side. It's completely gone. That's it. And next thing, the okay, uh, the mitochondria, the matrix of the mitochondria, like they have the infoldings, that is crista, and for respiratory chain reactions occur there. Okay, and crista has increased the surface area. Outer membrane also have the specific enzymes. Now the matrix of the mitochondria possesses single circular DNA molecule, few RNA molecules, and ribosomes, that is 70s, and the components required for the synthesis of the proteins. Okay, also the mitochondria present more in dividing cells. Chloroplast. So thylakoids, these are flat membrane sacs. Okay, and it has lumen. And chloroplast contains small double stranded circular DNA molecules and ribosomes. Okay, in endoplasmic reticulum, smooth endoplasmic reticulum, rough endoplasmic reticulum. Now rough endoplasmic reticulum, that is frequently observed in the cells actively involved in protein synthesis and secretion. Now they are extensive and continuous with the outer membrane of the nucleus. That's it. Its sister knee in Golgi apparatus. Now Golgi apparatus, disc shaped sacs or cisternae, right? Concave is trans maturing phase, and convex is cis, that is forming phase. Now Golgi apparatus do packaging materials, packaging of material is the main principle. Okay, and Golgi apparatus remains in close association with the endoplasmic reticulum. Okay, it found near the nucleus. That's it. In cell wall, there is middle lamella. It is a mainly of calcium pectate and magnesium pectate. and this middle lamella, it's a cementing layer between two cells. And now it's a plasmodesmata. It is for transportation of fluid between two cells. It is a kind of cytoplasmic threads between two cells. That's it. Now the line is the cell wall and middle lamella may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cells. Endoplasmic system contains endoplasmic reticulum. Endomembrane system contains endoplasmic reticulum, Golgi apparatus, vacuoles, and lysosomes. That's it. Now lysosomes, suicidal bags. These are like all types of hydrolytic enzymes for digestion of biomolecules and biomacromolecules. Schwann, it is German zoologist, work tells plasma membrane in animal cells and a cell wall in plant cells. And it's written that Schwann proposed the hypothesis that the animals and plants compose of cells and products of cells. And Sheldon, it was Schwann, now Sheldon, it is German botanist. Now he examined the plant cells. That's it. Now what is plasmid? In addition to the genomic DNA, that is a single chromosome, that is circular DNA in bacteria, right? Many bacteria have small circular DNA outside the genomic DNA. These are smaller DNA, these are plasmids, right? For phenotypic characters, that is resistance to antibiotics. That's it. Now plasmid DNA is used to monitor bacterial transformation with foreign DNA.